Hello everyone, welcome to my YouTube channel Microbiology Simplified. I am Dr. Amit Kumar Singh, working as professor. So again, a new video of my parasitology lecture series. This is lecture 7. It is about the life cycle of protozoan parasite. This is a very important video. Because why I am saying it is important? Because if you know the life cycle of a protozoan parasite, you will be able to answer so many questions. Because in the life cycle itself, so many details are present, like morphological forms of the parasite, the mode of transmission, the host, the method of reproductions. So if you remember, if you understand the life cycle, I believe that you will be able to understand the parasite itself and will be able to answer question asked on it. Also, lately I have seen that image-based question has been asked very frequently, and image-based question on life cycle is very difficult to crack if you don't know how to crack the information provided in the life cycle image. So this video is about the life cycle. So let's see that how I'm going to help you to understand this topic. I'll try my best. You try your best. Let's start. So just a quick introduction that what are the type of life cycle. So life cycle are of two types: direct life cycle and indirect life cycle. Direct life cycle are basically those life cycle in which there is only one host, single host. Okay. An indirect life cycle, which we call when there is two or more hosts. That is, number of hosts is either two or more than that. So then that is called as indirect life cycle. Okay. So usually if the host is single, the host is human host. And if the host is two or more in number, so one of the host will be definitive host and another will be intermediate host. So just if you remember, definitive host is those hosts in which we can see the adult form or sexual method of reproduction. Intermediate hosts are those hosts in which we can see the larval stage or asexual method of reproduction. Okay. So just now see that what are the key points you should know to identify the life cycle. Okay. So just by seeing the image, how you will be able to identify the life cycle. So you need to know certain key points or key information provided in that image. The first information which is usually provided are the morphological forms. So if you remember the various morphological forms, so any morphological form you can find either cyst or trophozoite or like merozoite, scisons, sporozoite, oocyst, anything. Okay. The host, you have to see the host, what are the hosts mentioned in that. Then you have to see the mode of transmission, what mode of transmission is mentioned, either it is ingestion, inhalation, or through insect vector, or through ingestion of contaminated ingestion of undercooked meat. Okay. Then you have to see the infective stage and the habitat of that parasite. Okay, that is what part of organ is involved in that parasite and what is the infective stage mentioned in that. And then you have to see the method of reproduction, that whether the reproduction is sexual or it is asexual or it is both. So these points, 5 points you have to see in the life cycle. The morphological forms, what are the morphological forms are there, what are the host, single host, double host, multiple host, mode of transmission, ingestion, inhalation, insect vector, infective stage, the habitat, which organ is involved, and the method of reproduction, that is sexual, asexual, or both. So I'll go one by one the life cycle of the various protozoan parasite and I will tell you that how to identify that. Okay. Before doing that, I would like to just make a quick recap of these 5 points of various protozoan parasite. So just go through it. I believe that this portion you may not be able to see because, so I will just tell. Okay. So in case of Entamoeba histolytica, okay, the morphological forms seen are cyst and trophozoite. Host is single host, that is human. Mode of transmission is fecal, and the infective stage is mature coordinated cyst, and the habitat is large intestine, primarily cecum, and the method of reproduction is asexual. Okay, so now you know these informations. So will you be able to crack the image of the life cycle of Entamoeba histolytica? So let's just see. So this first one. What are the morphological forms you can see here? First morphological form which I can see is the trophozoite, and second is cyst. Also, if you see the cyst, that is from this immature to this mature, what are the changes occurring? First, the number of nucleus increases from 1 to 4, and these structures, which are basically the chromatoid bars, disappear from the mature cyst. So this is typically the characteristic morphological forms of Entamoeba histolytica. So just mention it here. But we can see here trophozoite. What characteristic we have seen in the trophozoite? It is amoeboid without any flagella. Okay, and then the cyst, in which the immature cyst has got 1 to 2 nucleus, and the mature has got 4 nucleus. Okay. Now the second information, that is the host. What is the host here? We can see that only one host. Only one host we can see. One host, and that host is what? That host is the single host is human. Single host. What does it mean? Direct life cycle. What else we can see? What is the mode of transmission? Is ingestion. Okay. Mode of transmission is ingestion of mature cyst, which may be present in contaminated food and water. And what is the infective stage? Is obviously the mature cyst. And what is mature cyst? It is quadrinucleate. If you see the image here, mature quadrinucleate cyst. And what are the method of reproduction? So here we can see that trophozoite is multiplying into a trophozoite. That means through binary fission. The method of reproduction is through binary fission. That means asexual. Okay, so on the basis of this information, what we can derive? It's very simple now. What it is? It is which parasite? It is Entamoeba histolytica life cycle. So if you remember this, the morphological forms C, which can be seen: cyst and trophozoite, host, single, human, mode of transmission fecal through ingestion of contaminated food and water containing the infective stage, which is mature quadrinucleate cyst. And what is the habitat? Okay, I forgot to mention about the habitat. The habitat, if you see in this diagram, is large intestine. So the habitat of this is large intestine. So it is very easy to narrow down to Entamoeba histolytica. Okay. And the method of reproduction seen is asexual, that is binary fission. So it was the life cycle of Entamoeba histolytica. So now coming into the next, that is Naegleria fowleri. I will come to this table later on after discussing the image. It will be easy for you to understand. So what morphological forms we can see in this image? So just see, the first form we can see is the cyst. Second, first we can see cyst. Okay. Second form, then there are two forms which is changing its shape from each other, that is from trophozoite to flagellate, and then again from flagellate back to trophozoite, changing, exchanging the size, sorry, shape. So what are these? These are trophozoite, one is amoeboid form, one is flagellated form. So they can change their shape from one form to another form. So whenever they have to transfer from one water body to another body, they will change to flagellate, and then if they have to infect someone, then they will change to amoeboid. So how many hosts we can see in this image? Only one host, that is human. Single host. Okay. Direct life cycle. Mode of transmission. So just see, the mode of transmission is water related activities such as swimming, underwater diving and water sports resulting in entry of water in the nose. So it is through inhalation of the infective stage through nose. Okay, the mode of transmission is inhalation of the infective stage through nose. Okay, inhalation of infective stage through. And what is the infective stage in this case? It is the amoeboid form. Just see what is mentioned. The amoeba penetrate the nasal mucosa, the amoeboid stage, that is the trophozoite stage of amoeboid stage of trophozoite. Amoeboid stage. And what is the habitat? Although they are free living water bodies, but inside the human they causes meningoencephalitis. Okay, they causes meningoencephalitis. So their habitat is freshwater bodies, not free living water bodies, as I said by mistake. They are free living and they reside in fresh water bodies. Okay, but they causes infection of CNS, and the infection which is primarily caused by this species is, as mentioned in this image, is primary amoebic meningoencephalitis. And the method of reproduction, if you see, it is asexual. There is no sexual stage seen in this. So where we can narrow down from this information? Say trophozoite, two stages, amoeboid stage and flagellated stage. Single human host, inhalation, water bodies, inhalation of the infective stage from the water bodies. That means it is about a free living amoeba. Why I am saying? Because of the stages mentioned in the image. So it is about the free living amoebas. So what it could be? There are three breeding mummies: Naegleria fowleri, Balamuthia, and Acanthamoeba. So the disease is primary amoebic meningoencephalitis. By Acanthamoeba and Balamuthia, the disease caused is granulomatous amoebic encephalitis. They causes granulomatous amoebic encephalitis, that is Acanthamoeba and Balamuthia. They causes granulomatous amoebic encephalitis. So what it could be, what is the name of the parasite? The parasite is very obvious now. It is Naegleria fowleri. Okay. So this is the way to extract the information and come into the conclusion. Okay. So just go through the table. Naegleria fowleri, cyst and trophozoite. There are two forms of trophozoite: amoeboid and flagellate. Single host, that is human, mode of transmission inhalation. Trophozoite, cyst can also enter, but the infective stage is trophozoite, and they reside in the freshwater bodies and reproduce asexually. Now coming into the next image. Now just forget about whatever I told you in the previous all the lectures and just see that what is mentioned in this image. In this image, what we can see, if you don't know anything, I'll solve this without having any information about the parasite. Just see that whether I'll be able to solve it or not. Okay. So what we can see it is mentioned cyst. Okay, cyst is mentioned, trophozoite is mentioned. So these two things are mentioned here. And in trophozoite, we can see this structure. So okay, these are the flagella. Okay, so let's count the flagella. 1, 2, 3, 4, 5, 6, 7, 8. Okay, so the number of flagella are 8. 8 flagella I can see. And the nucleus in cyst is 4 nucleus. 4 nucleus, if you remember, it is quadrinucleate cyst. Okay, this is Entamoeba histolytica. But wait, Entamoeba histolytica is amoeba. They don't have flagellate. This has got flagellate. So what other parasite which has got uh, 4 nucleus and has got flagella in the trophozoite stage? Okay, I don't know. Let's see what other information given. So the host is only one host I can see, that is human, no other host. Okay, and the mode of transmission which I can see is through contaminated water and food containing the cyst. The infective stage is cyst. And it is causing disease of small intestine. Okay, so small intestine, especially the duodenum and jejunum I can see, not exactly, it's mentioned in the image. This upper part of small intestine I can say. So what it could be? So if I have got some idea about the name of the protozoan parasite itself. So what are the protozoan parasites? Amoeba, flagellates. Okay, flagellates. Flagellates are of two types, if I remember. Lumen dwelling flagellates and one is human flagellate. So this is intestinal. So it is a lumen dwelling flagellate. Among lumen dwelling flagellates, most are non-pathogenic except Giardia. Okay, so this may be Giardia, and as it is causing infection of upper intestine, so it is Giardia duodenalis. Oh yes, if you remember the number of flagella present in the trophozoite of Giardia, it is 4 pairs of flagella, and you can see this bilateral is symmetrical. You can see the 4 pairs of flagella. So this is the life cycle of Giardia duodenalis. Why I have done like this? Because in examination you may not be able to recall everything, because you have to study so many subjects. Microbiology is not one of the not the one subject, only subject you are going to uh, study during the preparation. You are going to study 19 different subjects. So for that you have to develop an approach which required less things to remember and more concept. So that's why I told, just give an example how to crack the question just on the basis of slight information. This whatever I recalled was a very minimal information about the protozoan parasite. Even if you remember the minimal information, you may be able to crack the question. So for that you need to have your basics clear. Okay, so just see properly that what are the morphological forms we can see here. First is the cyst, and how many nucleus we can see? 4 nucleus. And the other morphological forms which what we can see here is trophozoite. And in this trophozoite what we can see? 4 pairs of flagella and 1 pair of nucleus. Okay, that is total 8 flagella and 2 nucleus. Fine. And the host is again single host, that is human. So direct life cycle, and mode of transmission is ingestion of contaminated food and water, that is through fecal oral route. And the infective stage is this cyst, the mature cyst, and the habitat is small intestine, especially duodenum and jejunum, that is upper GI. And the method of reproduction what we can see here is asexual. It is only transforming from one form to another form and multiplying by binary fission. There is no sexual multiplication occurring here. So what it is life cycle of Giardia duodenalis. Okay. Coming to this next image. So what morphological form we can see here? Okay, only one form, and that is mentioned trophozoite. Okay. So fine. I'll derive the informations. Morphological form seen trophozoite. No other form. Trophozoite here, trophozoite there, and multiplying by binary fission. Okay. So reproduction is by binary fission. That is again asexual. And then the mode of transmission is mentioned here is sexual intercourse. Host again only humans. And infective stage is obviously there is only one morphologic form, so infective stage is only trophozoite. And habitat, you can see here it is pointing toward the genital organs. Okay, so it may be vagina or it may be the urethra, because the entry of the male genital tract is through urethra. Okay, so just write, note it down. In examination, or you can also note down the various points or information given in the image and then try to answer the question. It may take time, but definitely you'll be able to answer correctly. So morphological form seen here is trophozoite, and this trophozoite, just a little more detail. How many flagella you can see? 1, 2, 3, 4, and then this undulating membrane, flagella over undulating membrane. So there are total 4 flagella which are anterior, 4 anterior flagella, and 1 flagella running along undulating membrane. So they have total 5 flagella. Okay, and they are uninucleate. These trophozoite are uninucleate, and no other stage, no cyst. Okay, host human, single. Mode of transmission P to P, person to person, and what is the usual mode is sexual transmission. It's mentioned here in the question. Infective stage, obviously there is one stage, so infective stage is Propozoite. And habitat is, in case of female it is vagina, in case of male it is urethra. And the method of reproduction seen here is asexual, binary fission. Okay, so what we can conclude on the basis of given information: what is the pathogen, what is the protozoan parasite? This is Trichomonas vaginalis. Why I have not mentioned this information? Because this information may or may not be present in the examination. So better to try to understand the images rather than going through the text, because sometimes they may remove the text from the question. So it's better to understand the image provided. Okay. So one more solved. So now let's see the next image. Okay. So now we can see now the number of hosts is increased, the morphological forms, so many things. So start with the morphological forms which we can see here. The first morphological form which we can see here is this promastigote, and then this amastigote. The promastigote transforms into amastigote, and amastigote then multiply, and then it is coming out again only amastigote, which again transform to promastigote and then divide the gut and migrate, and then again. So only two forms. One is promastigote, another is amastigote. Host. So now I can see there are two different hosts. One is human, and then there is an insect. Okay. So insect vector. So there are two hosts. Two. One is human and one is insect. So let's see whether they have mentioned the name of insect or not. Okay, they have mentioned the name sandfly. Even if it is not mentioned, don't worry. You will be able to crack the answer without even knowing the name of the insect vector. Mode of transmission, obviously through insect vector during blood meal. And the infective stage, you just see it is mentioned. Even if it is not mentioned, just see the image of the morphological form. So what morphological form it is? It has got a flagella. This does not have flagella. And there are only two stages. One is amastigote, one is promastigote. The name itself suggests mastigote means flagella. So amastigote means no flagella. So what this stage is A or pro. Obviously promastigote because it has got flagella. Okay. And what is the habitat where it is residing? Inside the human body. Anywhere is it mentioned? Or in diagram can you see? No. But what you can see that it is entering in our body through blood. And if it is not mentioned the target organ, but we can understand that it may be present in the blood or in certain tissue. And the reproduction method what we can see is only transformation from one to another form and then multiplication by binary fission. So again, asexual method of reproduction. So on the basis of this information, can I answer the question? So let's see. So what I know that amastigote form and promastigote forms, these two forms together are present in only one protozoan parasite. None other protozoan parasite has got both these forms. Amastigote may be present in another protozoan parasite, but both these amastigote and promastigote together can be present only in one, that is Leishmania. So what are the other points which can confirm that okay, this is definitely Leishmania? So if it is mentioned sandfly, fine enough, because Leishmania, the insect vector is sandfly. If it is not mentioned sandfly, then how to derive? So see for the mode of transmission through Obviously it is through insect vector, so these will be present inside the blood. Okay, then chances are very high that this parasite is causing infection of the reticuloendothelial system. And also we can see this promastigote is the infective stage. So this promastigote is again the infective stage is only in leishmania. Okay, and there is no other method of reproduction other than asexual reproduction. So we can conclude that this is the life cycle of leishmania donopeni. Okay, so by this way you can identify the life cycle, and just don't only try to identify the life cycle. This is video is not how to identify the life cycle on the basis of image. This is also in this video you can see that in a single image you are getting so many information about the life, about the morphological forms, about the host, about the mode of transmission, about the infective stage, about the method of production. So try to remember all these things together. In leishmania, what we can remember: morphological forms, promastigote, amastigote, host to human insect, mode of transmission through insect vector, infective stage promastigote, habitat blood, method of reproduction asexual, and what is the insect vector which is actually transmitting the disease is sandfly, and what is the name of the species scientific name? Phlebotomus. Phlebotomus, and the common species is Phlebotomus argentipus. Okay. Okay. So try to extract the information which we can get from the image. So if you just remember the image itself, you will be able to remember half of the information about the protozoan parasite. Now coming to the next image. The next image what we can see, okay, slight overlap, so don't worry. This is the morphological forms, this is the host, this is mode of transmission, infective stage and reproduction. So what information we can get? Here the morphological forms. So see, first morphological form mentioned is metacyclic tripomastigote, then tripomastigote, then again it is taken up by blood meal by a fly and transform into procyclic tripomastigote, and then again apimastigote, and then to tripomastigote. So these are the morphological forms. First is metacyclic tripomastigote. Okay. Second is tripomastigote. And then procyclic tripomastigote and apimastigote. Okay. So these are the morphological forms which is shown in this image. Okay. What else we can see? Any difference between these morphological forms, apimastigote and tripomastigote? So just a brief review. This tripomastigote, this from where the place the flagella is originating as an undulating membrane before coming out from the anterior end, is posterior to the nucleus. This is the nucleus, and this kinetoplast, this is posterior. So it is tripomastigote. Whereas in case of apimastigote, this is anterior. This kinetoplast is anterior. Okay. So this is the basic difference between apimastigote and tripomastigote. And also if you remember this apimastigote, or even if you don't remember, you can see in the image this apimastigote, metacyclic tripomastigote and procyclic tripomastigote, these morphological forms are seen only inside the insect vector, that is the fly. These are not seen inside the human. Inside human we can see that this immediately this metacyclic tripomastigote, once it reaches the bloodstream, will transform into tripomastigote. So tripomastigote forms are seen in humans, whereas the rest of the stages are seen in insect vector. So this is only seen in humans, and these two and this one in insect vector. So how many hosts we can see in this image? One is human and one is insect. And what is the name of the insect vector is fly. Scientific name is Glossina. Okay. And what is the mode of transmission is bite of insect vector. And the infective stage is metacyclic tripomastigote. And the method of reproduction, again, we cannot see any sexual stages. So there is no gamete, no gametocyte. So that is no, says nothing. So this is, again, the method of reproduction is asexual. So on the basis of this information, that is on the basis of the host, the vector, the morphological forms, and the method of reproduction, what we can derive here, that this is the life cycle of trypanosoma. Okay, life cycle of trypanosoma. What it is, brucei or cruzi? Brucei. Why I am saying brucei? Because if it is cruzi, so if you remember the previous image, what I told you, the amastigote forms are present in Trypanosoma cruzi. So in this image, I cannot see any amastigote form. So that is why this is a life cycle of Trypanosoma brucei. Very easy to crack. If you remember certain things, then you will be able to crack the answer. So if it is mentioned, amastigote forms are also seen, and case looks like a case of a Trypanosoma. So then it is Trypanosoma cruzi. And if no amastigote forms are mentioned, then it cannot be cruzi and leishmania. It will always be Trypanosoma brucei. Okay. So as I told you, this is the life cycle of Trypanosoma cruzi. So you want to crack? Okay, just do it quickly. Morphological form scenes, as I told you, the first one is amastigote. The second form, what is seen is tripomastigote. Third form seen is epimastigote. Any other form? Yes, is metacyclic tripomastigote. Okay, so these four forms are seen in this protozoan parasite, and the first two are seen in the human host, and these two are seen in the insect host. Okay, so obviously there are two hosts, one is human and one is insect, and this insect host in this case, as mentioned here, is tritomine bug. This is also known as reed-wheat bug. And the mode of transmission, obviously, bite for this insect vector. And the infective stage, if you see here, even if you don't, if it is not mentioned, you can answer just by seeing the position of kinetoplast and the nucleus. When kinetoplast is positioned posteriorly to the nucleus, it is tripomastigote, characteristic of trypanosoma. And the tripomastigote form which is seen in insect vector is metacyclic tripomastigote. Okay, just see the image. Metacyclic tripomastigote. Okay. And what is the habitat? Again, it is in blood. And what is the method of reproduction? Again, there is no sexual stage seen, it is asexual. So what we have derived on the basis of the information, it is Trypanosoma cruzi. Very easy. Amastigote form, epimastigote, trypomastigote. If it is amastigote cruzi, if there is no amastigote, only trypomastigote or metacyclic trypomastigote and apimastigote. Apimastigote and trypomastigote are characteristic of Trypanosoma. And if amastigote is also included, it is Trypanosoma cruzi. No amastigote, Trypanosoma brucei. Okay, remember it. And if it is amastigote and promastigote, leishmania, very easy to crack hemoflagellates on the basis of morphological form itself, even if you don't remember the name of insect vector. If you remember the morphological forms, you will be able to answer it. So again, I told you, just don't learn these lectures only to crack the images of life cycle. What information you got apart from the morphological forms, the host, the name of the host, the mode of transmission, the infective stage, and the method of reproduction. So all these information you are getting. Okay. So okay. So what is this? So many cycles: human liver stage, human blood stage, mosquito stage. So many stages are there, and then so many different morphological forms are also seen. So just write small because so many forms I can see here. The first one which I can see is the cyzones. These cyzones are present inside the liver, and from this cyzone, what is released are the merozoites. Okay, and this merozoites, and these merozoites develop into trophozoite, initially immature trophozoite which form the ring stage, and then mature trophozoite which is amoeboid in shape. And this trophozoite further develop into cyzones. Again cyzone, but this cyzone is extrahepatic in the blood, and this is hepatic cyzone. This cyzone contain a very less number of merozoites compared to this cyzone. Okay, and then again this ruptured cyzone will release the merozoite. So again this, it may go into this cycle, or this merozoite may develop into, again now you can see gametocyte. That means sexual reproduction is occurring now. The gametocyte, male and female, which is then taken up by the insect vector during blood meal, and then this gametocyte will further develop into gametes, microgamete and macrogamete, and form ookinetes after fertilization, which will develop into oocyst, and this oocyst contain the sporozoite which is released, and then these sporozoites are released into the blood during the blood meal of the parasite. So this is the life cycle of parasite, and what I can tell on the basis of this, what parasite it is. It's not very difficult, because once you will see that so many life cycle and so many stages, you may be able to crack it just by seeing the life cycle image itself. But okay, just follow the protocol and see what information we have gathered from this image, and also try to remember the other important features of the of this protozoan parasite. So what are the morphological forms? See, one is hepatic, cyzont. Okay, then merozoites are released from it, then it develop into trophozoite, which further develop into cyzone, which is erythrocytic cyzone. And then again merozoites are released, which develops into gametocytes, that is male and female, which again develop into gametes, micro and macrogamete, which forms ookinete, and then oocyst, and liberates sporozoites during blood meal. So these are the list of morphological forms seen in this life cycle. What are the host? There are two hosts. One is human, one is insect. And which insect is mentioned here? Anything is mentioned here? Mosquito. Okay, the name of the mosquito is not mentioned. Okay, whatever. Mosquito. And what is the mode of transmission mentioned here? Bite the mosquito during blood meal. Okay. An infective stage, if you see, it is the sporozoites. Infective stage mentioned here is sporozoites. And then habitat is blood, that is RBC, blood cells, red blood cells. And the method of reproduction, now till now we have seen the method of reproduction is only asexual, but in this you can see the stages, sexual stages. So the reproduction method is now both asexual and sexual, both. Okay, so on the basis of this information, what it could be, it is the life cycle of, tell me, the life cycle of Plasmodium. It may be vivax, malaria, falciparum, whatever, depending upon the morphological forms in detail seen in the peripheral blood smear. If it is mentioned the detail of the morphological forms, then you may be able to identify the species also. Okay, and what is the mosquito? Which is the definitive host in this case is female Anopheles. Okay. So is it easy or not? Yes, it is easy. If you understand the basic, you will be able to answer it. So just remember the morphological forms, the various stages. So on the basis of that, you will be able to answer the answer of the life cycles, and also you will be able to answer so many questions asked on the parasite. So let's move into the next. Okay, very little information mentioned here in writing. So just see the images. First, the morphological forms. What we can see, the tissue cyst and the oocyst. Tissue cyst and oocyst. What else? Host, we can see humans, we can see cat, we can see rodents, amphibians, pigs, goat, so many hosts. Human, rodents, pigs, okay, cattle, and then cat. Why I mentioned separately cat? Because if you see, the oocyst is only liberated by the cat and not by these other hosts, nor by neither by human nor by, these are also not releasing oocyst, these are also not releasing oocyst, and then humans also not releasing oocyst. So only the cat is releasing oocyst, and that nothing is mentioned about the method of reproduction. But oocyst, actually oocyst contains sporozoites, and then oocysts are produced by asexual method of reproduction only. So that means the cat in this case is the definitive host, because in this it is the asexual method of reproduction is occurring. And human, rodent, pigs, all these are intermediate host. And the mode of transmission, what I can see is ingestion of food and water contaminated with oocyst, or ingestion of undercooked meat containing the tissue cyst. Ingestion, fecal-oral. Food and water contaminated with oocyst, or ingestion of undercooked meat containing the tissue cyst, both are causing the infection. However, as humans and these other different hosts cannot produce the oocyst, so they are basically dead end host. Humans are dead end host because they cannot be dead end because they can be eaten, but human cannot be eaten by the intermediate definitive host. A cat cannot eat humans. So that is why these may be the uh, may not be the dead end host, but human are dead end host, and they can also get the infection. We can see here by blood transfusion. Okay, let me write this in a more dark color. So iatrogenically it can. So if you just want to know the various mode of transmission, you can go through the lecture of mode of transmission of protozoan parasite, which I made. Infective stage is oocyst and undercooked meat containing tissue cyst. So both are infective stage. And the method of production, as I told you, it is producing oocyst, which is produced sexually. So it has got both asexual and sexual mode of reproduction. Okay, so on the basis of this information, what we can derive, this is the life cycle of toxoplasma gondii. So what are the key features to identify this? First, the definitive host, that is the cat. Cat is definitive host in only one protozoan parasite, that is Toxoplasma gondii. Okay, and also the morphological forms, that is the tissue cyst. Tissue cyst is present in, apart from Toxoplasma gondii, in Sarcocystis. But then the definitive host in that case is not cat. It is cattle or pig in case of Sarcocystis. So we can narrow down by these information that this is the life cycle of Toxoplasma gondii. Okay. So now move into the next life cycle. Okay. So what we can see in this life cycle, the morphological forms which we can see here are okay. Start here. Okay. Oocyst releases sporozoites, forms trophozoite, type 1 meroont, merozoites, type 2 meroont, again merozoites, and then microgamont, macrogamont, microgametes, and then thin walled oocyst sporulated, and then thick walled finally. Okay, so these are the morphological forms we can see here. So just note it down. So what we can see, the first is oocyst, which contains sporozoite. Okay, oocyst which contain sporozoites. Then trophozoite, then type 1 and 2 meroont and merozoite, and micro and macro gametes. Okay, and what are the hosts we can see? Only one host, that is human, no intermediate host. And mode of transmission is mentioned here is contaminated food and water containing cyst. So during recreational activity we may either drink it or through ingestion with food. So fecal oral. Okay, ingestion of contaminated food and water containing the oocyst. So infective stage in this case is the sporulated oocyst, thick walled sporulated oocyst causing infection through ingestion, and autoinfection can also occur by thin walled oocyst. And reproduction, we can see here both asexual and sexual cycles. That means these gametes can indicate sexual cycles. So reproduction occurs both sexually and asexually, both. But these two method of reproduction is occurring in only one host. So this is the characteristic of this particular protozoan parasite. And what are these protozoan parasite? These are coccidian parasite, especially those coccidian parasite which causes opportunistic diarrheal infection in the immunocompromised individuals. Okay. And also mentioned cattle are the major host for Cryptosporidium parvum. So usually they will not mention this, because if it is mentioned this, then you will be able to answer without even knowing anything. Okay. So on the basis of the information, so what could be the common among the other coccidian parasite are the oocyst, trophozoite, the merozoite, micro and macrogamete. You may find these stages in cystoisospora also and cycloisospora, but this type 1 and type 2 merons, this type 1 and type 2 meron are only present in single species, which is, what is that? Which coccidian parasite has got type 1 and type 2 meron are Cryptosporidium parvum. Cryptosporidium parvum. So this type 1 and type 2 meron is the key point here in this image. Okay, it is only present in Cryptosporidium. So on the basis of all this information, it is very easy to narrow down to Cryptosporidium, because only coccidian among sporozoan, that is, who reproduce sexually and asexually both. That is, there are very few. That is the hematozoa and sporozoa. They are only, sorry, hematozoa and coccidia. These two sporozoan only multiply both by sexual and asexual method of reproduction. So among them, fecal route, so hematozoa immediately will get eliminated. Now coming to these toxoplasma, so then again you need to see the tissue cyst and an intermediate and a definitive host, that is the cat. So again that is eliminated. Now among these Cryptosporidium, cystoispora, cycloisospora, the morphological form which is characteristic of Cryptosporidium parvum is type 1 and type 2 meron. So if you see type 1 and type 2 meron, it is a case of Cryptosporidium parvum. Okay, and also just to add, these oocysts are acid fast and they can be stained by modified Zn staining by using 1% sulfuric acid. Okay, so this next image, if you see that the morphological forms seen are merozoite, then macrogamete, microgamete, and then oocyst, and then this sporozoites. So sporozoite which is released from the oocyst, merozoites, gametes, and then oocyst which contains this sporozoites. There is no type 1 and type 2 meron. Okay, fine. And then mode of host is only one, although sexual and asexual, but there is only one host. That means there is no definitive or intermediate host in this case. Mode of transmission is ingestion, fecal oral route of the infective stage, that is oocyst or spore related oocyst containing the sporozoites. And the habitat in this case, if you see the image, is small intestine. I forgot to tell you in Cryptosporidium parvum also, if you see this image, the habitat is small intestine. Okay, and the method of production is both sexual and asexual. So what it could be? Now it may be cystoisospora or it may be cyclospora. So how to differentiate now on the basis of this information provided, whether it is cystoisospora or cyclospora? Tell me anything? Easy. See the shape of oocyst. Cycloisospora. The name itself suggests the cyst is circular. Circular in cyclospora. Whereas it is elongated in cystoisospora. So what we can see here, this oocyst is elongated in shape, okay, and it contains two sporocysts, and we can also see each sporocyst contains 4 sporozoids, the total of 8. 2 sporocysts, the total of 8 sporozoid, which is characteristic of cystoisospora. Cyclospora contain 4 sporozoids in 2 sporocysts, whereas it contains 8 sporozoids in 2 sporocysts. That means each sporocyst in case of cystoisospora contain 4 sporozoids, whereas in case of cyclospora it contains 2 sporozoids. Okay, so this is again a very important differentiating feature between these two. So on the basis of the information given in this image, it is very easy to narrow down to that the life cycle belongs to cystoisospora. Okay, so I hope in this video I have given a lot of information about the protozoan parasite, and also given you a method to derive the life cycle, that on the basis of image itself you can tell that the image belongs to which protozoan parasite or the life cycle belongs to which protozoan parasite. So hope it may be a lengthy video, but definitely it is going to help you a lot, because in this video it is not only the life cycle which I have discussed. I have also discussed the morphological forms, the host, the mode of transmission, the infective stage, the habitat, and the method of reproduction. So even if you go through this video itself, you will be able to revise almost 60 to 70% about the parasite. Okay, so all the best of luck for your NEET PG examination. Take care. Bye bye.